Okay, here we are, Machine Learning and Cloud Services. Part 1, Part Subset E, Lesson E, Clustering in general with examples. Just a few general remarks from my experience with examples from some practical work I've done in the past. All right, so let's go for it. So as it says here, general comments and examples from my research, clustering in general. And as you do your projects, you might learn a little from what I experienced. And of course, we wrote WebPlotViz based on these lessons that we got. This slide I really made a mess of. I don't know why. I'm getting a bit tired, maybe. All right. So we have some different purposes for clustering. The first purpose is to take a set of points which are over here, or over here, or over here, and really separating them. That's actually when I studied clustering first. That's what we were doing. Because our data tended to come from measurements where there were distinct points, and the clusters were generated due to measurement error. The application, which is I think much more common in today's world, is you just have no clusters, just a mess. But you want to find points near each other. That's actually KNN is particularly designed to do that. But clustering can also do that, because I showed you in the previous lesson that you can take, when I did the dating set and divide it into 30 parts, well, those 30 parts were fewer points and they were nearer to each other than when you divide into 3 parts. There are related methods of clustering called mixture models and latent factor identification models, which are closely related to clustering, and you tend to add probabilities from with some underlying effects. They're called latent because they're not obvious, and they have very similar formula to clustering, and they are similarly used to group items together. These are how one does things like Google News when you do topic modeling, when you try to find all news items which have the same topics. And then we've already mentioned that we have 2 types of methods. Those that work in real vector spaces, real 69 spaces, and the points are in those spaces. They have scalar products which are well defined, and distances calculated by the usual formula for vector spaces. But then there are other spaces like that form when you use from DNA sequences where the sequences are not aligned. When you look at those sequences, unless you do so-called multiple sequence, proper alignment, which is extremely difficult and probably non-viable in the real world, those sequences are not aligned. They're not vectors, but they have distances between them defined by the Smith-Waterman, Needleman-Wunsch or related formulae. So this example here from the past is a case where there's clusters, real clusters. This is Lymphocytes, and Lymphocytes has real clusters, and pathology, 54 dimensions, does not have real clusters. These don't, but here, this thing here is a real cluster. This thing here is a real cluster. Whereas here, we've just taken a sort of egg-shaped collection of points with a little bit of dribble from the edge of the egg, and we chopped that egg shape into some number of sub-eggs or regions. And the sophisticated methods are useful over here for lymphocytes, but they're not so useful over here for pathology, because the pathology is inevitably approximate. You don't need sophisticated approximation. All right, this shows this pathology case where there's not so much going on. Here we have 8 regions, and here we have 100 regions, and they are all have the same feature. You just get smaller regions if you have 100 clusters than if you have 8. And I say it can still be useful. I mean, certainly Amazon finds these useful, because if you're bought by your blue frog, it wants to know what items are near that blue frog. So if it clusters the space of items that they're selling, then it will find out which items are near blue frogs, because if that's what these clusters all have the feature that anybody in the same cluster is near each other, and k-means guarantees that. The k-means algorithm is highly reliable in that regard. It doesn't get fake spread out clusters. All right. Then we have some more examples here of separated where they're real clusters and not so unknown. Some where there are not real clusters, where there are real clusters. Mass spectrometry is a real two-dimensional space. The genomic case, which we actually saw already in the web plot viz examples. And remember here where we are projecting to 2 dimensions, and whereas if we use web plot viz, we'll be able to view in 3 dimensions. And we already showed in web plot viz where things can lie on top of each other in 2 dimensions, but clearly separated in 3 dimensions. So a lot of my work comes from a project called Spidal, which developed powerful machine learning algorithms which had high performance and ran in parallel. When you start applying these problems to millions or billions of points, you really need a lot of computing, and you have to run the programs in parallel to be able to finish them. A lot of the work I showed you ran in parallel, not on huge machines, maybe 500 cores, but they were programmed quite carefully in parallel, so they actually got good performance, speed ups, near 500 on 500 cores. We've always emphasized that we've always found visualizations critical, which means we must have dimensional reduction. And we have found the results when you only have distances work equally well to those which are in vector spaces. They just take longer to do if you only have distances. And in fact, formally, the methods with vector spaces of order n, where n is the dimension of the space, when you have just distances, they're inevitably of order n squared, because if you have n points, the number of distances is n squared, and you have to calculate them. And it's worth noticing in those plots I showed you with the DNA sequences, it took as long to calculate the distances as it did to do the clustering or the dimensional reduction. So we have fungi data, and we had 7 million sequences originally. FASTA is the standard way of recording sequence data where you have 4 characters for each position in the sequence. And we first removed duplicate sequences, and then we applied the Smith-Waterman algorithm to generate 170,000 by 170,000 distance matrix, which we just store on disk and read in every time. And our program is called DAPWC for deterministic annealing, parallel clustering, and so in this case here, we start off with 8. We actually use the human eye to guide the clustering. We start off with a small number of clusters, look at the results. Sometimes take one cluster and just break it up, and that actually means we get a smaller problem. Rather than running the full data set, we break the data set up. And then we add up the results to get the final answer. So all of this took a long time. Life is quite hard. So here we have the 170,000. They're now joined back up, and you can see that they clear clusters. We showed that this is not the same data we showed, but like we have this clear red cluster here, a little green cluster here. Sometimes they're on top of each other as here, but we'd have to rotate them to see if they're really on top of each other. Some of these are on top and some are separated. Here's just a result of rotating. Notice the dust over here. Things that are on the, when you do these multi-dimensional scaling, things on the edge are sort of screwed up. And it puts on the edge things that are sort of don't fit well in the plot. They go on the edge of the plot. So here's just a third view. We have a similar type of structure, and again, clear clusters, not so clear clusters. Fourth plot. Here we have an interesting thing. This is a case where we had 420,000 clusters, and this is just a tiny subset of the data. There are 10 million points. The points are the small things, like we have a small green point here. The yellow are the center of the clusters, and the orange stars are just points which are not clustered. This particular case was the case we knew the measurement error, so we knew the size of the cluster, and that is forced by the clustering algorithm. So if you look at these results, you can see some, like this cluster here, is very clear. But here, we have a clump which is divided up into 4 parts, or 5 parts if you include this, or 6 parts if you include this. But that division is not so clear, and it's and you only see that really if you visualize it. That this data consists of clear clusters. It's suddenly very clustered, but when the clusters are relatively near each other, they're pretty uncertain, and different algorithms and different criteria will give you different clusters. So this is a so-called LCMS mass spectrometer data. We're finding so-called peaks, and as I've already told you, it's 11 million points and 420,000 clusters. And this actually algorithm, the 420,000 came from the program. We just actually started with one cluster, and it gradually looked in greater and greater accuracy of the data to find out the total number of clusters. All right, here's our favorite handbag formula picture, which we saw from WebPlotViz, and here are this comes from the COG database. These are the COG family already identified, and with a number of points in these biological clusters. And we've already commented that the blue and the reddish things are pretty clean. That there's a cluster of actually what the experimentalists felt was similar clusters over here. And some things like this white cluster are pretty spread out. This is a way you really have to know that you have a decent answer. So if you look at this here, what we've done is we plotted for the handbag problem the original distance here from the Needleman-Wunsch calculation, which calculated the distance between every 2 sequences. And here's the result. Then we project the Euclidean space and we plot the Euclidean distance versus the original distance. And so it's not too bad. I mean, in a perfect world, this would be a straight line along the diagonal. But this is a heat map, so when the color is very intense, that means there are a lot of points. So we're pretty near that straight line, and there's some distortion here. It's pushed away from the origin in the original data. And there's a little bit of a maybe it's not so important, a small distortion here. But then basically it's pretty good. Here is the actual distribution of distances in the original determination. I should say that Needleman-Wunsch, when you have distances, it's sort of pretty heuristic. You can choose new distances, and that is why it's called transform. We took Needleman-Wunsch and applied a transformation to it to actually spread the distances out. If you looked at the original Needleman-Wunsch formula, it would look more like, well, I don't know what the scale is, but it will be a much more peaked. And if it's peaked, then all the distances are the same, and so it doesn't get such good answers. But you can apply a transformation function to so-called reduce the mathematical dimension of the problem, and that gives you more spread out, and then you'll get a more reliable answer. Here's some interesting data. This is from the biologist, and they have labeled the data, and then we've visualized it. And so we've noticed here's some green points here, and the same green points here, which says the biologist thought these two were in the same sample, but they obviously aren't, because they're very distinct. So they're really two different species. Here we have white and white. So here, two totally distinct, well-populated sequences. Here, light green and light green, well, so grayish green and grayish green, and actually blue and blue. And they should really, if they should be actually joined like this and like this, not what they've done. They've chopped each of them in half. So this points out that visualization, not just of clustering, but just of any classification, could be very helpful because you can really see possible flaws. I note one feature of all these visualizations, they use colors. And now if you go to the color set of all possible colors, you cannot find hundreds of distinct colors. And probably you can tell all the colors apart on this plot, but there are only about 16 independent colors that the eye can easily tell apart. And so that's why if you look at some of my earlier plots, which have hundreds of clusters, the colors actually repeat because you can't find 420,000 useful distinct colors, and in fact you can't even find 100 useful distinct colors. So they choose a number like 32 or 16 and just repeat them, which means you can get two clusters with the same color when you look at it. So you just have to check. But I've checked in this case, and these are the same cluster, not the same color used for different clusters. All right, here's an example of the 170,000 DNA sequences, and they're clustered. And now we can imagine taking, as we will later on, take the centers of these clusters. Then we can compare the centers of these clusters with existing data of this type. And we've done that with the red squares. We mapped the red squares into the same three-dimensional space. And unfortunately, there's no resemblance between the red squares and the original data. So it says that these fungi come from Scotland, that maybe it's so cold in Scotland, or so wild, or too much haggis, or too much whiskey, that these fungi are pretty different from the original fungi plotted by the other experimental biologists. And this is shown again here, where we actually replace those clusters, which are sets of sequences, by their center, and then we plot those centers, which we actually use we distinguish supercluster, well determined. We have 211 clusters of the Scottish data. We have 124 of them are pretty solid. They're more than 200 members. 87 are not so solid. They have smaller than 200 members. But the yellow and the red and the original, which are now the original data outside the Scottish fungi, which are now white, unfortunately not the same color as the previous plot where they were red. There's no resemblance. So that just says that the Scottish fungi are just different. We also can do things with so-called phylograms, which is to take what biologists like to do and produce tree-like formulae to show how things evolve. And we can do that not just in 2 dimensions or 1 dimension as the biologists tend to do, but in 3 dimensions. And that is actually displayed by WebPlotViz. And there's the pictures here. So this is an old data set. Here is the actual formula, which has been the sequences, and it's visualized here. And you can see it's pretty nice. We have nice 3-dimensional trees. So this is sort of interesting. You can do tree-like hierarchical structures from your data. Notice here that these red things here should be clearly from the data distinct. But they're viewed as the same in this particular classification. I believe this data comes from the biological labeling. Here is this, we've already effectively done this. This is this 2-dimensional proteomics data. This was a small sample of 30,000 points. And in this case here, the clusters are pretty clearly clusters. There's not as much data here, and they're not nearly as much on top of each other. Here is a sort of perfect example. This is some test set of this is from the deterministic annealing pairwise, PW is pairwise clustering, and you can see it's been applied to this metagenomics data set, and I guess this was prepared as a test set, the test methods, and we are getting essentially perfect answers. Here is the real data from 500,000 data set points. Is a subset of 73,000, nearly 74,000 points. I told you I took the clusters and divided them up into 8 subclusters. Took the original data, divided them into parts, and looked at each part separately. This is one of the parts. This is work is with Hai-Shu Tang, who was the faculty at Indiana in bioinformatics. And you can see this cluster is pretty reasonable. It's found lots of subclusters, and we have 25 at the moment, and you could possibly get more because some of these things, like here, we have some blue clusters which might be split up. Here is data which is the original data, 440,000 data points, and it has been broken up into subclusters. The first it has actually 7 clusters, and these lines in the middle are just the coordinate axes, which happen to be displayed on this plot. We should remove it, and you can see that after we formed 8 clusters, it's actually given you a set of red sims, which you look at separately, blue things, light blue things, which are pretty separated. So this idea of breaking things up into large subclusters is actually quite successful. Here's one of those subclusters which we then look at in detail, the one which had 15,000 sequences in it, and you can see it has these tadpole shapes, which we already commented on, but otherwise it actually splits into clusters pretty nicely.